Good evening. Thanks for joining us, everyone. And it's been a heck of a couple of days here, hasn't it? And hopefully we'll put our minds on some different topic tonight. Those Americans who may be joining us from Canada, especially welcome. Tonight we're going to talk about genetic immune cell and microbial interactions in inflammatory bowel disease, celiac, and some other GI disorders. This is a huge, huge topic. I just came back from Harvard, where Alessio Fasano invited me to attend the 20th year celebration of the Celiac Treatment and Research Center. That's at Mass General, that collaborates with some other institutions at Harvard. And at that occasion, he and the other heads of the other centers that are collaborating in this area spoke a little bit about the past 20 years and what's transpired in this field, and it's quite a lot. We'll touch on some of that by way of review here. The thing that was also interesting is that they spoke about future research. And the future research that they see is this topic, genetics, immune cell interactions with the gut microbiome and the metabolome. And this is to say, therefore, that the topic it's a new horizon. It's extraordinarily broad. The findings that are coming out very, very quickly. And it'll be something that I think will guide medicine for some time to come. The purpose of this presentation is to give a little bit of a foundation to the basic concepts and some of the research that we've been involved with some of the institutions like Yale School of Medicine with respect to the food component of this equation, and see how the pictures emerging that puts these things into juxtaposition with one another and kind of make sense of it. The gut microbiome is it's like another organ system in a way. By the way, I just want to start here. I'll explain this in a moment. There's about 1,000 times more genes in the gut microbiome as there are in the human host. We have fewer than 30,000 genes in the human genome, but there's probably 50 times that in the microbiome. So they all interact with one another. We have a symbiotic relationship. The human provides a home and safety for the microbes, and the microbes provide services, provide the synthesis of short-chain fatty acids for foods, for enterocytes, and synthesis of vitamins that are used by the host, and also are involved in educating the host immune system, and they continue to interact with the host immune system, and that's kind of what we want to talk about a little bit here. What we do is to try and bring a framework to this, is offer some testing that helps clarify when a patient presents with gastrointestinal symptoms, which direction to go, which kind of diagnostic procedures to follow. Do they need a referral to a specialist? How likely or unlikely are they to progress to celiac disease? Do they have celiac disease? What are the interactions with foods and Crohn's? Why is Crohn's there? What can we do about it? There are things that we can do about it that are non-invasive. So we have an assay that we call the CICA, for short, the celiac IBS and Crohn's assay, which has a number of different markers in it. And some of those, as you see, are genetic markers for identifying risk, if a person is at risk for developing celiac disease. And then other genetic markers for looking at risk for Crohn's disease. And it's kind of interesting is that as we move along, and I'm going to touch on some examples, particularly with the Crohn's, some of the reasons that we thought those genes were risk factors for the disease are becoming expanded upon as we go deeper. And just recently, there was an interesting article in Science, just in July, that identifies a new role for these genes in the etiology of Crohn's disease. And I think as we move along, we'll dive deeper and see more of these things coming to light, and we'll understand even further about this very complex constellation of this interaction between the symbiotes, are the host, and the microbes. Of course, we can gain insight by looking at serological markers that can tell us whether or not there's an active disease process going on with celiac. And of course, we add to that as an option a cellular test, the ALCAT test, which we won't have time to review in much detail today, but this company, Cell Science Systems, which sponsors the Cell Science Collegium, is known for having developed this test. And I'll just mention a little about the company now in that regard. We've developed this test over a period of 30 years. And this Cell Science Systems is a laboratory, federally licensed and state licensed, but it's also a medical device manufacturer. And we manufacture the instrumentation for carrying out this cellular assay, as well as all the reagents and buffers that are used in the process. That means the food extracts and so forth, or we go to Whole Foods, buy the foods, bring them here, lyophilize them, and then process them and use them in the assay. And we do this all under a quality program that's supervised by the FDA and compliant with ISO medical device manufacturer standards, which keeps our certification. And that's good because we have some then more control over this process, and it allows us to really understand a bit more. We can run experiments very easily. We're fully independent and fully integrated, so we can run more experiments and understand a bit more about how the cells, the leukocytes, the peripheral immune cells, interact with dietary components and other environmental components. So we'll touch on that a little bit, especially since there's recently been some report about clinical findings and technical findings about the cellular measurements that the test exposes. The SICA report's easy to follow if there's, homozygous positive or negative. We have either a very low or a very high or an elevated risk or somewhere in between if we're heterozygous. And that's for both the genes that relate to celiac as well as the genes that relate to Crohn's. Let's focus in a little bit about what those genes are. We look at the human lymphocyte antigen DQ2 and DQ8 genes, as most people do. And I want to speak for a few moments about why we do that. What do those genes do? Why do we measure those? Just for an overview. So that'll give us a little bit of a basis for understanding the progression to celiac in those individuals that have that susceptibility, or why there is something called non-celiac gluten sensitivity. So this is an old, but I think very useful graphic that's borrowed from Scientific American from Dr. Frisano's article some years ago that he called The Inside Story. And it shows here, of course, the enterocytes, in this case, probably a damaged enterocyte that's kind of leaky. So we have paracellular transmission of cellular products as well as dietary products, in this case, gluten fragments that are able to penetrate into the lamina propria, where, we have 80% of the immune system is resident in the gut. And so there's a lot of stuff going on right here. So in the pathogenesis of celiac, which is what this graphic is showing, is the trigger of gluten fragments that can bind with tissue transglutaminase. So you'll notice when we look at the particulars of the of the testing that we do is that we measure antibodies against TTG, tissue transglutaminase, to see if it's present. Why? The clinical significance is that TTG is produced from damaged enterocytes. It then, it on the one hand indicates that there's a disease process going on. So if a person has a celiac genes but they don't have TTG, well, they still have to avoid gluten because they have those genes, and it could erupt if other cofactors present themselves, which could be microbes or other factors. So we want to look for the presence of the deaminated gliadin peptides, which we know are toxic. And of course, in this illustration, there's a leaky gut, so they have more access into the lamina propria and then into the general circulation. And they combine with TTG and become immunogenic, meaning that they'll be recognized by an antigen-presenting cell. That complex will be taken up by an antigen-presenting cell, most commonly a dendritic cell, and brought into sort of a lysosome. The peptides are broken down and bound to a molecule that translocates to the surface and then presents it to a T cell that will recognize that molecular conformation. And only a T cell that recognizes, and that's why we're talking here about the adaptive immune response, or specialized cells, the T cells. The mediator here, of course, is this molecule that picks up the peptides and translocates it. That's called a major histocompatibility molecule, or MHC, class 2 molecule. Now, if that interaction does not occur, which we see depicted here, the T cells don't get involved, or if they, or much less so. And then we don't have the progression of the cytotoxic T cell lymphokines causing the damage that we see here to the enterocytes. We may have irritation, we may have other disorders which we'll broadly characterize as non-celiac gluten sensitivity, which could give rise to symptoms that we call IBS, but we won't have that progression to celiac. And that's why it's interesting to look at the presence of those genes. If the genes are not present, the molecule doesn't exist, and the presentation of the antigen and the antigen recognition process does not occur. And so therefore, everything on this side of the graphic doesn't happen. We don't get the T cell communication to the B cells to produce antibodies. We don't get the activation of the T cells and the transformation of the T cells into cytotoxic T cells that then cause this damage. So that's why we test for, that's why we test the human lymphocyte antigens for those genes. Now, of course, the antibody test against the deaminated gliadin peptides has been documented very well, and sometimes different people, particularly younger versus older, and vice versa, will have more of the IgA isotype present and less of the IgG, and vice versa. So they should both be present in the evaluation. Looking ahead now just a bit to the other part of the equation, microbiome. When we start off, there's not that much of one, really. And of course, natural childbirth will help inoculate with beneficial, hopefully beneficial bacteria from the mother. A C-section will abrogate that. So when possible, natural childbirth, natural anything seems to have probable benefits to living organisms. And then there's a change over time in the composition. And there's really only 4 phyla that, that comprise the majority of the microbiome. Bacteroides and Firmicutes are about 75 or 80% of it. And that composition changes over time. And of course, it becomes much more individual over time. We have greater diversity and less individual variability as we age. So why do we care? Of course, we hear about it every day. As I was mentioned, at a minimum, the gut microbiome contains at least 500 to 1,000 different species. It's very, very, very diverse. And these guys, they can be friendly, can be commensal, they can be pathogenic, and everything in between. The microbiome composition will vary significantly in abundance and the type between individuals, within the individual, depending upon the location inside the GI tract. In fact, the types of microbes that tend to colonize in areas where there's inflammation are different. Now, we don't know if that's a cause or effect, but we'll find that out as time goes on. Purpose-wise, they help synthesize vitamins. They are involved particularly in carbohydrate metabolism. They manufacture short-chain fatty acids, which is fuel for the enterocytes. They help develop, they help the immune system mature, both locally in the gut and throughout the whole body. We've all heard now about the hygiene hypothesis that there's more atopy, there's more, meaning allergy and asthma amongst children that grow up in a more sterile environment. So the early inoculations that we experience not only during birth and breastfeeding, but throughout exposure to the environment in a natural way, help develop tolerance. Then, of course, commensals will, they want to survive like everybody else. So we have good bacteria there. They will help exclude the colonization or the expansion of colonies of pathogenic bacteria. And there's also other communication that takes place not only with the immune system, but also with the nervous system. So it's critical. It's like a whole new organ. In animal models, IBD, that's what we wanted to talk about actually in this presentation. They play a role. These microbes can be protective or they can be pathogenic. And it's all contextual. And contextual means how the immune system has been programmed and our genetic factors. And that's what I want to address in a moment. We know that when you manipulate the microbiome through a fecal transplant, sorry. We don't want to transplant fetuses, I apologize for that. It changes the phenotype. So that's not a surprise. Antibiotics have been shown in some experiments to reduce gut inflammation, but one must be cautious. Obviously, using antibiotics, it will drastically reduce, maybe by 50%. A course of antibiotics can reduce by 50% the abundance of microbes in the gut. An area we particularly want to focus on is that the microbes impact, not only we're talking about antigen presentation through the innate branch of the immune system or the dendritic cells, which are professional antigen-presenting cells that sort of sit in the middle between the innate and the specific immune system, as we saw in that graphic moments ago. But the microbes talk to the T cells. That not only influences T cell function and immunity, but also factors that impact the barrier function of the gut. And that's critical, and that's a very complex area that we should delve into entirely on its own because it is so interesting. So it leads us to conclusions that we have our genetics, but does genetics and even knowledge of the microbiome also, is that sufficient to explain phenotype or symptomatology or disease or health? No, it's suspected other factors are environmental, and of course that would primarily be diet. I mean, food is the biggest exposure that we have to the environment. Now, foundational to any understanding is that microbiome studies have shown that IBD, excuse me, IBD results from a dysbiosis. So what dysbiosis would that be? First of all, in looking at a review paper fairly recently, metagenomic studies have shown that in IBD, there's an upregulation or an expression of genes, an upregulation of genes that are responsible for oxidative stress pathways for inflammation, and a reduction in the expression of genes that are associated with normal metabolism. The reduction in bacterial diversity is a hallmark feature of IBD. Particularly, there's a decrease in Firmicutes, which is normally the most prevalent phyla, and particularly among in a reduction in Faecalibacterium prausnitzii. And here is something that, of course, is just a reference. We won't go through this right now, but in CD, also Crohn's, ulcerative colitis, we see in surgical biopsy, in stool, consistently in CD and UC, a reduction in the Faecali prausnitzii almost across the board. And then the others, they're kind of mixed, but that's a critical one. So that's one that you may want to look at. We don't do that. We don't look at the microbiome right now, but we have some assays in development, but there are labs out there that do have that. So, although the precise cause of IBD remains unknown, what makes most sense, or the most acceptable hypothesis, is there's an aberrant immune response against the microbiota, which can be triggered by environmental factors, meaning, to me, mostly food, and that in a genetically susceptible host. Now, we looked at the genetic antecedents that are associated with celiac disease. So let's look a little bit at the genes associated with Crohn's. Most of you might know that the Nobel Prize in Medicine this year was awarded to Dr. Osumi for his discoveries of the mechanisms of autophagy. Now, autophagy is a normal process that's very critical to homeostasis. Autophagy, as the name suggests, is the eating of self. And that means debris that comes from damaged organelles or other physiological components. It's like phagocytosis. When it's disrupted, it's quite a problem. You can imagine if you had human corpses lying around, they would spread disease. You don't, on the microscopic levels, you don't want that happening either. You don't want dead organelles hanging around. So that's an interesting perspective when we start looking at the disease antecedents, or the genetic antecedents associated with Crohn's disease. So here's two of them. And these are the markers that we test for in our SICA assay. ATG16-like 1, that's a gene which is associated with autophagy, formation of the autophagosome. NOD is one of the several classes or several types of pathogen sorry, it escapes me at the moment, the things that recognize repeat patterns on the cell walls of pathogens. So the NOD1 and 2 will sense, and that's intracellularly. We have these sensing molecules that can activate innate immune cells in defense against pathogens, both on the surface of the cells and on the surface of enterocytes, on the surface of immune cells. And they're both on the surface and also within the cytosol in the event that a pathogen penetrates into the cell. So the NOD1 and 2 are involved in sensing those repeat patterns molecules inside the cell. And what they'll do is they'll recruit the ATG16L1 to the site of bacterial entry, and that triggers autophagy. So in this review, these authors conclude that the close connection between NOD2 and ATG16L1 suggests the importance of this pathway in the pathophysiology of Crohn's disease. And that's why we test for those. Now, you can't explain it just by genes, because we have our genotype, and that may express itself or may not, depending upon epigenetic factors. And most of those factors, and they cover the whole range of everything that we think, that we breathe, that we come into contact with, that we eat, our internal physiology. So certainly what we eat is a major factor. But just actually in July, there was an article in Science that takes our understanding about these genes, these autophagy genes and these NOD-like receptor genes, to a deeper level to explain a nuance that wasn't understood in quite the same detail. And what that was is that the immune system, again, mediated by the dendritic cells, culminating in activity of the lymphocytes, the specialized cells, the T cells, is impacted by at least one commensal bacteria called Bacteroides fragilis. And what this microbe does is express in what's called its outer membrane vesicle a substance called PSA. Now, it's not important to remember what those substances are right now. Other than that, they have to do ultimately with recognition by the dendritic cell, communication then with the T cell in the same fashion that we saw just a moment ago. Again, the dendritic cell communicating with a T cell, but in this case, instead of a food particle, think of a microbe. I'm sorry that I don't have a graphic for that, so I have to make do with that. And that PSA that's expressed in by the microbe, and which is sensed by the dendritic cell, then communicates messages to the T cell, which causes the T cell to upregulate its production of interleukin-10, which is anti-inflammatory. So what we have here is a pathway where polymorphisms in the susceptibility genes promote disease through defects in sensing the protective signals from the microbiome. In this case, it's the PSA expressed by the Bacteroides fragilis through its outer membrane vesicles coming into direct contact with the dendritic cell, which then communicates with the T cell and kind of modulates the T cell activity in such a way that it inhibits or doesn't allow for the production of a very critical inflammatory suppressing cytokine, interleukin-10. So that's another model explanation of the pathogenesis of inflammatory bowel. And from that study, it's a wonderful study. You can look at that reference in Science. I recommend it. Just one of the illustrations here is levels of IL-10, interleukin-10, measured in the plasma of mice who are either normal, wild type, or have that mutation, the same mutation we're looking at in our assay. So what we can see is that with control, there's not that much difference. PBS is just a control just a buffer. But in the wild type mouse, the stimulus from the outer membrane vesicle causes a high level, relatively high level of the production of this beneficial interleukin-10. In the genetically impaired mouse, very little is produced there. So you can see if you short circuit that and deliver the PSA directly without going through that pathway to the T cell, you also get the production of the IL-10, which shows it's not the T cell that's defective, it's the pathway, which involves the dendritic cell. So what we're talking about are these genetic antecedents in the dendritic cell, which again is modulating the T cell. What can we do about this? Well, there's a number of things. Probiotics help. There's been a number of studies that show various probiotics are effective with inflammatory bowel diseases. And this is a good reference to look at. And they're equivalent to mesalazine in many cases, which is a non-steroidal anti-inflammatory, or on their own, they're maybe even more effective. So these are all the studies that are looked at for the different, this is a combination of probiotics. Now, of course, probiotics require prebiotics. And prebiotics, I'm just going to pause on this for a moment. Prebiotics, of course, can be given as supplements, but the best prebiotics are the right kind of foods. And the foods that nurture the beneficial commensals are things that contain a lot of fiber. And in fact, the Firmicutes, which is very beneficial, has been shown to be represent a greater preponderance of the microbiota of children who eat it's called a natural diet, children in villages in Africa, versus bacteroides that will predominate in children that eat diets that are high in refined foods. So the fiber is the best prebiotic we can use. And we can use supplements as we want. We can do both, but I think the diet is the best way to go if you can do it. I want to segue now just a little bit into some of the research that I think is going to help set some new directions for understanding the effects of food on the immune system, or the interactions between food and the immune system. This is a paper presented a couple of years ago, and I suspect it's probably too small to see the reference here, but it's on our website. It shows that there's an upregulation of a particular molecule on the surface of T cells when there's exposure to a positive food. And CD11B is a molecule which is an adhesion molecule. And if we won't do it again because we've looked at a lot. The graphic, we'll just keep that in mind as we move forward. Just let's keep that one in mind. So CD11B is expressed more on immune cells when there's exposure to an ALCAT test positive food. More recently, just this year, there's been a constellation of studies that are done, technical studies done at Yale at the Digestive Diseases laboratory there, that show a couple of interesting findings. One finding is that the positive foods or the cellular response is associated with certain mainstream immunological pathways, and we've looked at those, and the particular ones that are involved are protein kinase C and NF-kappa B. And they're related to each other, so that makes a lot of sense. And of course, NF-kappa B, probably people have heard about it a lot in the context of chronic inflammation. It upregulates, it translocates from the cytosol when it gets activated, migrates to the nuclei and causes the upregulation of various inflammatory genes. So that is taking place. So we can now begin to look at beginnings of a framework for when we're looking at gross changes in cells as we do in the ALCAT test, that there are understandable pathways that we know result in inflammation being activated. One of the other things we see is, as indicated by the title, is that a positive food is associated with increased levels of DNA fragments in the supernatant of the sample where the positive foods in the cells have interacted, much more so than a negative sample. And that's significant because obviously the DNA, whether it's coming from the mitochondria because that's been damaged or the nuclei because that's been damaged, doesn't belong outside the cell, it belongs inside the cell. And when it gets outside the cell, it creates problems. And we'll go into that in just a moment. And which cells are involved? Well, there's probably a number of different markers that can be looked at to determine activation of cells. One almost universal activation marker of inflammation is called CD63, and that's almost always upregulated in eosinophils. Now, eosinophils are cells that are associated with allergy. Micrograph of an eosinophil shows there's the nuclei and a number of different granules that contain various chemicals that are either inflammatory or non-inflammatory, like IL-10 or eosinophilic cationic protein that can be, or EPO, that can be very, very inflammatory. And if we look here at what the eosinophils do and why we say they're associated with allergy, allergy is sort of our, now we're talking about classical allergy, not delayed allergies, not a food sensitivity, or we're talking about a type 1 hypersensitivity reaction. That's our defense against worms. And of course, I hate to be this poor guy. I mean, these eosinophils are just ripping him apart and not digesting him. So that's something that I think kind of gives us a little bit of insight. What's going on when we see in an ALCAT test cells getting activated? I'm going to go out on a limb here for a second and say probably when you see cells getting activated, you're looking at something that shouldn't happen. The immune system's there when it needs to be there to protect against danger, but you don't want it getting triggered, every time you have a meal. Let's go back for 1 second. This was from a study from Harvard, from Fasano's lab, in cooperation with some of the labs in NIH looking at lymphocyte biology. And here we see, this is a section of the gut from an experimental animal that's been exposed. And in this case, they're exposing it to gliadin, pepsin, trypsin, gliadin, so it's pre-digested the way it would be if it gets down to the gut. And comparing the cellular response, sorry about that, to, the cellular response to a control, and also a known experimental substance, FMLP, formylmethionine leucine phenylalanine, which is a peptide that occurs on the walls of bacteria. What we're seeing here is the activation of these neutrophils in a transgenic mice, a mouse, where they've been treated so that they will fluoresce, so we can see what's going on. And after a little bit of time, they're kind of exiting from the small vessels into the tissue. So here again, think about the Yale finding of the upregulation of CD11B, which is allowing for that process to take place. So we're beginning to see a picture emerge there. This is what it looks like in vitro, where we're looking at FMLP in this panel, which you'd expect to start activating neutrophils, causing migration, gluten versus a control, where we don't actually see that taking place. Here we go again. So again, gluten is causing, at least ex vivo and in vitro, which we've seen in the previous slide, activation of cells. Now, is it only gluten that can cause the activation of the innate immune cells and lead to these issues that may or may not advance to activation of specific immunity, or just stay as a sensitivity, like non-celiac gluten sensitivity? Here's an interesting video, and this comes from a clinic in Kiel. And this patient's presenting with, he's got a lot of problems, but you can see he's got some skin problems, periorbital rashes and so forth, and he's talking about mainly his terrible GI problems. They investigated this guy through blood tests and scratch tests to look at reactions to common allergens, and the antibody test and this in vivo test, the scratch test, didn't show anything. So they did something very experimental. They using a probe, they're actually looking for the, and we'll see this in a moment, the response of the enterocytes or the gut to the presence of direct injection or direct exposure of different common food allergens. These are in German, of course: milk, soy, wheat, and yeast. They're injecting a radio contrast media so that they can, with a light, see which substances, when they're exposed directly into the gut, into the large bowel, are producing a greater transmission of light, kind of analogous to TEER, decreased electrical resistance, in other words, greater intestinal permeability. And they're trying soy, they're trying milk, they've tried wheat, and no result yet. But when they get with the yeast, for this patient, of course we're all different, they see the screen light up. So we see that breakdown in the intestinal permeability, in this case because of the yeast. Now, it's interesting because yeast is a living organism. Now, of course, we're not necessarily testing it while it's still alive, but it still has that conformation, just like the FMLP. And we know that, it's not shown here. I apologize, I don't have a reference for that right at the moment. But we know that antibodies against yeast are associated with Crohn's disease. And that's why we also include in our seeker assay, our gut assay, anti-Saccharomyces cerevisiae antibodies. Now, focusing back again on the same constellation of studies from Yale. I apologize, this was in German. Because of the election, I didn't have the opportunity to translate it back into English. But we see here that there's a much greater release of DNA from the cells when exposed to an ALCAT-positive food versus no food or a negative food. And in their presentation of this poster, the investigators say that this is the first demonstration that specific food items, and these can be, and these are food items other than gluten and yeast, which of course are biggies, but it could be anything, can result in release of DNA by peripheral blood leukocytes, and further, these items can be identified by the ALCAT test. This finding may provide a mechanistic rationale for the reported findings of clinical improvement in patients using the ALCAT test. And further research should be warranted. Now, let's put that in perspective. There's a lot of studies now that show that when cells, neutrophils, and other granulocytes, other innate immune cells, including monocytes and macrophages and eosinophils, become activated, one of the strategies that they can implement in a little bit of a later phase is the production of what's called an extracellular trap. If it's from a neutrophil, it's a neutrophil extracellular trap or a net. But these nets or extracellular traps are produced by other granulocytes as well. And they'll trap pathogens. And what do they contain? They contain proteins and enzymes from the cytosol, but also chromatin and DNA particles from the nucleus that merge with the cytosol and then form these, along with the membranes, both inner membranes and cell surface membranes, form these net structures to trap the bacteria. And the DNA is actually quite toxic to the bacteria. That's all well and good. What happens to the debris? Well, we have an enzyme system that will clean that up. DNase 1. But what happens if there's too much of it? We don't have enough of that enzyme. Or if we don't produce enough of that for genetic reasons, then we have too much of this DNA fragmentation and chromatin circulating around. And it's been shown to be a major factor in inducing autoimmunity, particularly lupus and RA. And here's a nice report that just came out in July called Nucleic Acid Targeting Pathways Promote Inflammation in Obesity-Related Insulin Resistance. So when you have obesity from diet, it actually also promotes excess release and diminished clearance of nucleic acid. So it also promotes that, but it also is a side effect of that. The DNA actually will induce metabolic problems. So you get into this cycle, as well as the generation of autoantibodies. So we don't want that DNA floating around outside the cells. And to make it even more complicated, and I'm getting close to the end right now, is some research that comes out of School of Public Medicine at Harvard, where they've identified something called 6-transmembrane protein of prostate 2, which was first discovered in the prostate, which exists in the membranes of the adipocytes. And it provides a counter-regulatory mechanism so that when you have a high-fat diet or high-carbohydrate diet where you naturally promote inflammation, this counter-regulates that. But you know what? If there's a genetic deviation, and at least in the case of experimental animals, it hasn't not been demonstrated in humans yet. But when you have STAMP2 negative mice, they don't have STAMP2 in their visceral white adipose tissue, in their adipose tissue around the tummy, you get problems with transport of glucose. And that's also expressed in the other adipose tissue, the subcutaneous. So we're beginning to see kind of a pattern emerging here that there's a very, very, very intricate and finely tuned balance between our genes, our exposures to the types of food, the macronutrient composition of the food, the individual components of the food, our microbiome, and our immune system. And it is so complex, and that's why I think the people at Harvard have identified it as being the focus of their study for the next number of years. And just to conclude here, also in the experimental mice, in the visceral adipose tissue and in the subcutaneous adipose tissue, the animals that are, these are the wild-type mice, but the animals that are lacking the STAMP2 genes, they don't have the STAMP2 molecules, they produce a lot more inflammatory cytokines and also haptoglobulin. And it's interesting, haptoglobulin 2 is zonulin, which causes gut permeability. So there's a lot to think about in these findings. And I hope that's been helpful, and we'll open up to questions right now.
First, thank you very much for that very engaging presentation. We are at the top of the hour, so we'll just keep it to 2 questions. First, for those healthcare providers who are, getting exposed to this topic, perhaps for the first time, can you recommend any good resources for those who are, enthusiastic to dive deeper into the topic?
Yeah, there's a lot. There's Nature Immunology just had several review papers and published, and I've saved those, and I think they're open access. And if that's what you want to do, that's what I'd recommend. Shoot me an email, and I'll send them to you.
Great. And maybe we can include that in the follow-up to this presentation.
Absolutely.
Second was a question with regards to T-cell signaling and antibiotics, and if you're aware of any research or science that might show that that antibiotics interfere with the T-cells signaling capability.
None that I'm aware of off the top of my head directly, but obviously antibiotics are really going to change the composition of the gut microbiota, and that's going to impact on the cells that modulate T-cell function. So indirectly, there will be many of them, and there's probably so many different pathways. And again, that'll be something else that's contextual and in the light of the person's genetics and other environmental exposures. So I can't say there's anything specific that I'm aware of. But again, the approach is kind of holistic. Let's eat the right diet and avoid the food triggers or other environmental triggers that we know cause aberrant immune reactions. And the right diet means certainly one that's healthy for the microbiota and compatible with our unique genetically determined biochemistry.
Great. Well, I want to thank everyone for attending tonight. I want to thank you for your presentation. And for those who want to catch this again, we will be providing this, the video, transcript, and slides of this presentation in about a week from today. Thank you very much. Good night.